Hello, this is Group 8 of Section 10 of LB 144 Lab. I'm Noah Morrison.
Keaton Sayre.
Sydney George.
I'm Colin Harvey.
And our research project is the impact of highways on the biodiversity of Flora. So the inspiration from this project comes from the Berlin Wall and the Iron Curtain over Europe. In 1961, the wall was not only an imposing political figure, but also a geographical one, which separated up much of the landscape. In the areas around Berlin, deer used to migrate through where the wall was, and after the wall's presence and its fall, deer have consistently gone around the wall. This got us thinking about how large man-made structures affected wildlife and ecosystems around them, specifically highways in the United States. Habitat fragmentation, is a very real concern, and a paper by Aguilar et al. Discussing the genetic consequences of habitat fragmentation found that its long-lasting effects can be overwhelmingly negative. So our research question was, how do Michigan interstates affect biodiversity of plant species in an ecosystem as measured by the Simpson's Diversity Index compared to non-highway impacted ecosystems?
Okay, so now we're going to be talking about the knowledge gaps. So what we do know versus what we don't already know. We have predicted based on previous studies, including those mentioned in an article published by Ecology and Society titled The Effects of Roads on Animal Abundance, that biodiversity along the side of the highway will be impacted in a potentially negative way. While we do know how highways impact the biodiversity of animal populations, it is less clear to which extent highways impact plant life and how it could further impact overall ecosystem diversity. Okay, so now we'll be talking about the significance of our research. We believe that this project may have farther-reaching implications on more than just local Lansing highways. A marked decline in plant biodiversity due to the presence of highways has repercussions higher on the food chain. As plant ecosystems collapse due to lower genetic diversity, insect and animal spheres that subsist from those plants collapse as well. With enough evidence supporting the decline of biodiversity due to habitat fragmentation from highways, a push for heightened conservation efforts and funding by the government to protect native plant species could be achieved. As well, knowledge of how highways impact our environment can lead to more informed and more eco-friendly developments in the future. Okay, and now we'll be talking about methodology.
So the independent variable we're looking at is the environment, so whether it's a highway or by a non-highway, which goes to the dependent variable, which is the number of plant species and the type of plant species. We use that in the Simpson's Diversity Index and put that into the statistical analysis for our data. The Simpson's Diversity Index is a ratio between the number of plant species and the given amount of each of those plant species, where D is that diversity index value that you get, big N is the total number of species found in the environment, and little n is the total number of individuals found of a species in the environment. And the confounding variables are distance from the highway, general climate, and age of the highway. For distance from the highway, we looked at an article called The Effect of Intensive Traffic on Soil and Vegetation Risk, Element Contents as Affected by the Distance from a Highway, from the Czech Academy of Agricultural Sciences, which showed us that highways have an effect on the soil, which also affects the plant life at the highway. And to control the distance from the highway, we are measuring it by the tree line, so there isn't interference from cars, and is also away from the median and the side of the highway, where states purposely do their own landscaping. To control the climate, we're all doing the highways at similar longitudes because they're all in the East Lansing area. They're separate locations in different highways, but still in the same area. And to control the age of the highway, since all of the Michigan highways were completed by 1992, there have been 20 years at least for the surroundings environment to take shape as a result from the highway. For procedures, we used a quote from the Journal of Wildlife Management Guidelines for line transect sampling of biological populations. They emphasize that it is crucial to conduct observations along straight transects and obtain accurate measurements, distancing, and sighting angles. It is most important to conduct the survey in such a way as to maximize the probability that all objects on the center line are found. We are doing that by measuring along the tree line. For our procedures, we identified the roadside and rural tree lines ahead of time, and then we measured out 100 feet along the tree line with a tape measure, indicating the 10 transects. We used flags on every 10 feet for the transects. Then we take a picture of each transect and take a video of the whole 100-foot section. Then later we analyze the video data and marking different plants in the pictures.
Okay, so some measurements that we have to take. Like Keaton said, there is little n and big N. Little n is the total number of organisms for a particular species. Big N is the total number of species. Both of those numbers are very important for the Simpson's Diversity Index, which we will be using to measure the overall plant diversity for both transects. And we'll be using 100 feet for each transect divided into 10 feet spaces. Statistical analysis that we're using the t-test, which will give us a value which determines the significance of the difference in biodiversity between the two groups. Our null hypothesis is there's no significant difference between the biodiversity of highway and non-highway environments. Alternative hypothesis is there's a significant difference between the biodiversity of highway and non-highway environments. On the 30th of October, we went out and we did a sample collection test run. On the left, you can see two flags. Those flags are 10 feet apart, and you can see me on the right there. This test run was very good for us to have, as it allowed us to make changes to our overall procedure.